Hi, this is Miss Litton, and this is 7th period support for AP Biology, Chapter 13, Regulation of Gene Expression. Say hi.
Hi.
Are you sad?
Yes.
Are you overwhelmed?
Yes.
You can do this. This is a masterable quiz, I promise you. Look at you crying. Do you have a big test tomorrow? No? Oh, it's a long week. Okay, just take a big breath in. Take a big breath in. Let me try one more time. Hi, we're here to review. Can you say hi?
Hi.
That's the best you can do?
Yeah.
So I'm the only one working hard here. You can do better? Yeah. Let's be engaged. Let's make use of this time, okay? So the first thing we need to remember are all the steps that are taking place, right? So we've got DNA replication taking place if we're going to do cell division, either mitosis or meiosis. Okay? If we want to get that genotype out into a phenotype, we have got to do transcription. We got to get it into an mRNA and a functional mRNA, a mature mRNA if we're a eukaryote. And we got to get it translated into a protein. What's universal about that is the enzymes. The enzymes that you use to get from the DNA to the RNA. What you use to do translation out on the ribosome. But there are little steps that are different whether you're a prokaryote or a eukaryote. There are control mechanisms that are a little bit different, and that's what we wanted to study. Because we know cells have differential gene expression, which means that different genes are turned on and off in different cells and at different times, right? And at different times as well. So we looked at areas, first of all, for gene expression. The whole reason we did this slide is just to see that it can happen in multiple different places, that you can control gene expression all the way from is the DNA available for transcription and translation to what do you get when you get the mRNA, how do you need to process it, can it even get out of the nucleus, can it access a ribosome for translation. So we looked at all of those. Our introduction to it was looking at something simple, and that's looking at a prokaryote and its gene expression. So we reminded ourselves that they're not going to make any enzymes that they don't need. It's going to serve a purpose, and they need the amount for whatever job they're going to do. And so we identified there are some areas on the gene itself that are binding sites. What are, on this gene, what are binding sites on this gene? Promoter. What likes to get on the promoter? RNA polymerase likes to bind to the promoter. What's another binding site? Operator. What likes to get on the operator? The repressor. Is a repressor always on an operator? Is a repressor always born ready to repress? No? Because sometimes you need a what to make it so it starts repressing? A co-repressor, right? Remember co-repressors? You need something to get it to move on there. Okay, so we have promoters, we have operators. We learned more later, right? What are some other binding sites we learned about later when we talked about eukaryotes and even prokaryotes? A place, yes, enhancer in eukaryotes where activators bind. And we learned there are places where transcription factors can bind too that really usually neighbor the promoter. Okay, so I just want to remind us there's more to it than that. All right, so the first operon that we want to discuss is the lac operon. Now the lac operon is all about lactose. Remember, operons are a controlling regulatory region and all the genes that they do control. So for us, you're going to have your promoter, because the promoter is where RNA polymerase binds. You're going to have an operator where potentially a repressor can bind, and then whatever genes you're trying to control. In this case, 3 different genes that, if transcribed and translated, will make you an enzyme to digest lactose. Okay, that's what you're after. But if there's no lactose present, you don't want to turn this gene on because it's just wasteful. So the repressor that you have, by the way, where does the repressor come from? Yes, if the regulator gene is transcribed and translated, it will code for a repressor, and this repressor is good to go. As soon as it's built, it's like boom, and it's on the operator. And that's okay. Remember, prokaryotes often work in negative control. They turn things off until you need to turn them on. But that means you've got to have a lot of repressors doing that repressing job, right? We have way more DNA than prokaryotes. Way more DNA. So we don't work with negative control. What do we tend to work with? Positive control. We work more in the lines of positive control, which means you need several different components all agreeing, yes, we must go forward. Similar to what we studied with our immune system, right? The B cells and T cells all have to agree before you can move forward to develop an immune response. All right. So lactose comes into this bacterium because we ate it, and then the bacterium absorbed that milk sugar. Now, this repressor is going to bind to the lactose, and when it does that, when it binds to it, it lets go of the operator. Remember when we talked about enzymes and allosteric changes? This is an example of an allosteric change. Because the repressor is changing its shape, it can no longer bind to the RNA polymerase. The fact that this is here changes everything else. If I could make this animated, maybe these are some other shape here at the end, so it changes their shape as long as the lactose is there. Well, that's great, because the repressor's gone. Now RNA polymerase can bind to the promoter. It can start moving down here and then start transcribing these genes right here, and you make some mRNA. That mRNA is translated, and it's translated into a protein, and the protein that you end up making will digest lactose. Digest, 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 digest. Now there's nothing causing a conformational change, so the repressor again is available to bind to the operator. Okay, a very elegant way to turn a gene on and off when you need it. Now, we know, because we did there's more to the story, okay, we know there's more to the story on this. What's the more to the story on this one? What? Glucose, okay? When you have plenty of glucose, okay, you do not want to digest lactose even if it is available, okay? Because you would prefer to metabolize glucose. It's a monosaccharide, send it right through cellular respiration. Okay? But if your glucose levels are low, you start to have an elevation of what molecule? Cyclic AMP. When cyclic AMP increases, it binds to a molecule called CAP. CAP is a transcription factor. CAP will assist RNA polymerase to bind to that promoter. If you do not have CAP helping RNA polymerase bind to the promoter, it won't bind. And then transcription will not take place. And that's where we had those several different scenarios where we laid out and we said, would this transcribe, would this transcribe? The only way it'll transcribe is if glucose levels are low and lactose levels are high. Okay? Does that make sense to you? Okay, you're going to just jump to that right there. Okay, that was the lac operon. And then we also looked at what's called the trip operon. No, I don't want that one. I want this one. Okay, the trip operon. Now, in this case, with the trip operon, you don't have a repressor that's good to go at the very start. This particular repressor is, when it gets transcribed and translated, it is an inactive repressor. Okay? So that means in this case, this operon is on and it is repressible. Repressible, right? Not currently repressed, but it could be repressed, right? If you are an inducible operon, your operon is what? Off, but it could be turned on, exactly. So in this case, RNA polymerase is binding to the promoter. You don't see anything in here about a transcription factor, but RNA polymerase is binding to the promoter. The structural genes are being transcribed and translated, and your product that you are generating is tryptophan, okay? When you have enough tryptophan, you don't want to waste your time building any more tryptophan, so that tryptophan itself acts as a co-repressor, activating, again, causing an allosteric change in your repressor, right? It's going to change its shape if it's binding to tryptophan. And when it does that, it makes that allosteric change in shape. It will now bind to the operator, inhibiting RNA polymerase from binding to the promoter. This is very typical in activities that are anabolic, where you have a product you're trying to get. The product itself shuts off the process. If it's a catabolic activity, when it's present and you need to digest it, then you make the enzymes to do that catabolic reaction. Okay? Remember, positive control has to do with facilitating the process of transcribing those genes, and anything to do with a transcription factor is positive control. Negative control has to do with the repressor. Regardless of whether the repressor is currently active or inactive, a repressor is all about saying stop, and a transcription factor is all about saying what? Go. That's its purpose, right? That's its purpose. So look at what the purpose is to understand whether it's positive or negative control. Okay? So this is a picture of that cyclic AMP, and if you recall, we said cyclic AMP will bind to CAP, an inactive transcription factor, make it active. It'll bind to your CAP binding site, which will assist RNA polymerase to bind to that promoter. Okay? And then we went and looked at it. If you have an inactive CAP, yeah, you don't have an inhibitor here on your operator. There's no inhibitor, but RNA polymerase binds weakly, and so you don't get a lot of product as a result of that. And we looked at a diagram where you went back and forth and thought that through. I would be comfortable with those kinds of conundrums. If you have this and this, what's going to happen? If you have this and this, what's going to happen? You know, and be familiar with that. Okay? Then we leveled up and we said, okay, that's prokaryotic control. Let's talk eukaryotic control. So in eukaryotic control, we said there are so many areas where whether or not you have the product, it could be influenced. And the biggest one of all, what did we say was the major area of control? Transcriptional control. Okay, so of all of these areas, you can look for places where some sort of control will be used as part of a normal process of maintaining homeostasis. But you got to remember that if your control is not kicking in till the very, very end, then why did you waste your time going through all of this when you get to a protein and then you're like, nah, I'm not going to fold it correctly, okay? Because you've wasted your time, energy, enzymes, amino acids. So where you really want to hit that control is right here at transcriptional control. When you look at areas where your environment might be tricked in or turning on genes or turning off genes, that's when you're looking at things like epigenetic inheritance and chromatin structure, and which genes are unwound and expressed. I had a set of twins come up to me today in class when I was talking about sex-linked traits. They know another set of twins where one of them is colorblind and one of them is not colorblind. And the first thing I asked is, are they boy twins or are they girl twins? Now, you should be able to explain, no problem, while they were girls and they were identical twins, one of them was colorblind and the other one wasn't. And that had to do with the what?
Bar bodies.
Yes, those bar bodies, those X chromosomes. So for one, primarily, and they had a father who was colorblind, so that makes sense, right? Because the father every single time is going to give a bad X. And for one of the twins, that was the X chromosome that was expressed, and her mother's X chromosome was not expressed for her. That's why she's colorblind, whereas her identical twin sister is not colorblind. Transcriptional control, remember, we're going to focus here not so much on what we're repressing, but what we're turning on, what transcription factors are available, because you've got so many genes scattered across such a wide range of chromosomes, you can't possibly repress everything and turn it on together in that way. It's easier, like in this environment, when this, and this are present, then that works together to turn on those particular set of genes so you get that product. Post-transcriptional control. When you look at that area of control, it's not so much about whether or not a gene is expressed or not, but all the what? Variety. Sorry, that's not a good way to explain it to you, but It's all the different variety that you can get. That's where that control is. It's not like, oh, that gene's on or that gene's off. Post-transcriptional control has everything to do with exon recombination. How many different protein products can I get from the same gene? And our big example there was the immune system. Yes?
How do we know like what intron is going to come out? Is there some way there that codes for it?
You know what? That's where the research is. 30 years ago, the year I started teaching was the year they started or 32 years ago, was the year they started the Human Genome Project, where they were trying to map out, and we're going to talk about that tomorrow in class when we finish up this chapter, but they tried to map out every single nucleotide that you would find, generally speaking, in a healthy human, what that sequence would be. Not variety like brown hair, blonde hair, but all your important enzymes and structures. They try to map all of that out. They thought that would be the holy grail. If we know that, then we know everything. But there is so much that takes place with your post-transcriptional control, translational control, post-translational control in the environment of the cytoplasm. You can't, just because you know the DNA, doesn't mean you're going to get that protein. Because the folding pattern that takes place, what amino acids are snipped out in the process of the folding pattern has such a big deal. It's those proteins that are probably more important. And when we talk about introns and exons, a lot of there's intragenic and intergenic. There's genes between genes and genes that are in the grouping of genes. Those probably influence even more control than the genes themselves, because they're maybe coding for something that's going to fold it or some other sort of signal that works together in order to modify that particular protein. So yeah, that is what's trying to be understood. In order to make, let's say, a drug that you want to have in your body, like people will say, oh, just take your vitamins. You go to take a vitamin when a pill, oftentimes your body doesn't absorb it in that fashion, so it doesn't do you any good. It has to be this version, this isomer of it. Just because you have the sequence of amino acids doesn't mean you'll get the same end result, because it's not folding into the same pattern. Yes?
Do introns also choose which exons are expressed?
Do what? Oh, it can influence it. If they're methylated, if they're tagged, all kinds of things like that. That's the part that we don't understand, that we don't have really control over. Yeah. Okay? So translational control, you want to be concerned with like diseases and things where you have small interfering pieces of RNA, because if all of a sudden they're taking your great messenger RNA that you've made and making it double-stranded, then all of a sudden there's no place for your tRNA to land. tRNA can't bring that amino acid, whatever it's bringing. Okay? And then post-translational control, this right here, this is called proteomics. This is where a lot of study is taking place, because they're trying to understand the conformational structure of the proteins that we have and how they get folded. Those chaperone proteins are huge to get it into its functional shape. Okay, so just a few details on that. Things like epigenetic inheritance, or things are what's methylated, what's not methylated, how can our environment influence what DNA is unwound. We looked at bar body as an example of that. We looked at lamp brush chromosomes as evidence of genes that are actively getting transcribed. Also, we looked at heterochromatin versus euchromatin. If it's heterochromatin, you know it's put away and it's not getting transcribed. It's staining darkly. If it's euchromatin, it's light, it's not staining well, and that's probably because it's getting transcribed. Okay? And that can change within the day, within the week, within the month. Okay, our transcriptional control, we have the basics, we understand all of that. When we level up, we bring in transcription factors and how they influence it. We looked at things like that TATA box, right, where additional transcription factors can bind in order to help RNA polymerase bind to the promoter. This is something that's universal. This helps us go to our idea of a common ancestor, because that is a regular place where a promoter needs to bind. But we know, where a transcription factor needs to bind. But we know it's not just a matter of the transcription factor, but all of the other activators that have to be there to help that transcription factor get bound on. So the transcription factor itself could be kind of similar or universal, but whatever turns that on is where you could have your variety in that as well. And we decided it gets really more complicated than that to have everything in the right place. We also brought up the idea of transposomes. Transposomes are jumping genes. This is the DNA excising itself like a spliceosome. It's removing part of itself and inserting it somewhere else. Now, if you're looking at this from Darwin's point of view, you might say, well, that could increase what? Yeah, variability. This is how new genes could have arisen. So maybe it's not a bad thing. Yeah, unless it takes a gene that was a proto-oncogene and makes it a oncogene. Then you got trouble. Okay? And we looked at Indian corn as an example of that. Post-transcriptional control, cap, tail, cutting out the introns. We looked at, so the exons could be expressed, that you could have a piece of DNA and multiple proteins coming from two or three genes on the DNA just based on exon recombination. I showed you like hideous lists of different kinds of RNAs that could promote or interfere with the process of transcription and translation. We looked at translational control, your ability to get onto the ribosome, the lifespan of the mRNA, how long it lasts, how long it's available for translation. And we looked at post-translational control, and that was everything like we mentioned earlier about that folding pattern. Okay, then we looked what happens when things go bad. So one mutation you want to be able to recognize is a point mutation. And the way I remember that is you can point to the nucleotide that's causing the problem. Sometimes it doesn't actually cause a problem because of so many options for some ways to like code for proline or something like that, that it's called a what kind of mutation? Silent. Yeah, it's a silent mutation because you still ended up with the same product that you were after, tyrosine, because there was more than one way to get that tyrosine. Okay? A nonsense mutation is when you get a stop codon. There are 1 start codon, 3 stop codons, and if you all of a sudden have a mutation, a point mutation, which causes a normal codon to become a stop codon, then you're not going to complete your chain of amino acids. An example of that is hemophilia. Remember, hemophilia is you're missing a clotting factor. There's multiple clotting factors. You just need to miss one of them, and then all of a sudden it interrupts your ability to make a blood clot because you just don't have that one protein. And then a missense is you made a mistake and you put the wrong amino acid in that spot, and as a result of that, may or may not cause folding problems, and then it may or may not be lethal. Those are point mutations. Sometimes point mutations don't cause a problem. Sometimes they cause a little bit of a problem. Sometimes they cause massive problems. One that will always cause massive problems are frameshift mutations because you're reading all your codons and all your codons are off. And that can happen, if you said to me, should I delete one nucleotide or 3? What do you think would be the better thing? Yeah, delete 3. Maybe the one you delete, maybe you don't need that. Maybe that normally gets cut out anyway, right? But if you delete 1, all of them are messed up, right? If you delete 3, you're just short 1 amino acid. I think the odds are better that it's still going to work out, okay? Because you need to get the reading frame back into the correct spot, okay? We talked about how non-functional proteins can affect a whole pathway. Like if you're missing just this enzyme, you have PKU, you're missing the next one, you're also an albino. We also mentioned that there are some, genes that are not expressed. We've talked about that before, about penetrance and expressivity or a faulty protein. In this case, you have faulty receptors, so you've built a faulty receptor protein, so as a result, you cannot tell that testosterone was even present. Okay, and then we circled back around to cancer, and we reviewed the difference between tumor suppressor genes and proto-oncogenes, which can become cancerous. Those would be mistakes in your DNA that have long-term consequences on the phenotype. And we looked, P53 was your example of tumor suppressor, and RAS is like stepping on the gas. And then the last thing we looked at was our ability to detect chemicals in our environment that could be cancer causing. And it is not intuitive initially to think how they do it, but they'll take something that is pre-mutated and see if it unmutates. It's the only way I can, that way you can tell. Because you can't always see a mutation in a phenotype. It might be in the genotype, but you can't see it in the phenotype. So if you take something and purposely take a mutated version of it and it corrects itself, then you know it has the ability to change the DNA, and if it could make it go one way, it could probably make it go the other way, and that's not something you want to expose your body to. Okay? And that was it. Questions or concerns? Anything you don't get? Need me to go over again at all? Yes?
Is an example of epigenetic inheritance DNA methylation?
Yes, absolutely. Real quick, we have a few minutes left. I can't remember the essays off the top of my head, but I would imagine that I would ask you to compare and contrast a couple of things. Okay? I would ask you to compare and contrast an inducible versus a repressible operon. Okay? So I'd expect you to be able to tell me, you know, which one is used in a catabolic versus an anabolic, and why does that make sense? I would expect you in that situation, if I asked an essay on that for your exam, that you would tell me, you would have to set up the operon to begin with, you know, and you would tell me regulator genes code for repressors. Repressors can be active initially or need to be turned on by a co-repressor. You know, I would expect you to be able to tell me what each thing was. The promoter is an area where RNA polymerase binds to know when it should start the transcription process. And it doesn't start right at the promoter. It has to get past the operator, right? Okay. So you have, yeah. Okay, and then I would expect for you to tell me about the operator. I would expect you to tell me about the structural gene. Okay, and then compare and contrast the way those work, just like we did in class today. Okay, another compare and contrast would be positive versus negative control. Okay, and a good example of both of those. That would be a good essay question. Positive versus negative control. Okay, another question that I would ask is, how can you use positive and negative control at the same time on an operon? And what would you be thinking about with that? What? The glucose lactose, right? Okay. Another thing I would compare and contrast is prokaryotic versus eukaryotic control. That would be another good essay question. Another one I would ask is, in eukaryotic cells, there are 5 levels of control. Identify those 5 levels and give me an authentic example in each one of them. That would be very typical. I might ask you to compare and contrast a point mutation versus a frameshift mutation. Yeah, and their impact. Yeah, what they are and their impact. I mean, I might ask you about cancer again, but I'm really just kind of tying it back so you see if the DNA is messed up in these spots. So those would be critical areas where you would not want to have a mutation, right? Because you wouldn't want to mutate from a proto-oncogene to an oncogene or something like that. Okay, anything on that? You feel like you could answer those essay questions? Any questions on them? No? Okay, yes.
I was wondering if you could explain how the Ames test works.
Yeah, the Ames test, what they'll do is they'll take some bacteria that they have purposely introduced a mutation into, like the inability to make histidine or something that it needs, some component that it needs to survive. The inability to digest glucose, something, or metabolize glucose, something where that has a mutation that they can easily tell. And what they would have to do is they would have to take, and whatever they can't synthesize, you know, like our essential amino acids, those are the essential amino acids are ones we can't synthesize from any other amino acid in our body. All the other ones we can. Okay, these we have to actually eat. So they would take something that normally it would be able to synthesize that, and they would mutate it so it can't. So they start already with a mutant. And the only way that bacteria could survive is if you grew it on a plate that was embedded with that nutrient that it could no longer make. Okay, so that's what you're starting with. Okay, then you take that bacterium and you expose it to whatever chemical you're testing, and you're wondering, I wonder if this chemical does bad things. You would, once you exposed it, then you would put it on a plate that does not have any medium enriched with any histidine in it. If some bacteria, bless you, can grow on that plate, that tells you it had to mutate in order to grow on that plate because it couldn't normally. Okay, so, and it's just like one metabolic switch you're asking for a change. You're not asking like for some big evolutionary steps. It's like you mutated it, switched it off, now all of a sudden it's back on, right? So it mutated to form that. That tells you that that chemical is a mutagen, meaning it can change your DNA. Because if it can change this back, it could change something else forward into a mutation. If it is a carcinogen, it also causes what?